Okay, hello everybody. Welcome back to the TICAtalks. For our second TICA talk today, I'm Isaac Mark. I'm a graduate student at UC Berkeley. And I'm just gonna make a couple of general announcements. And if anybody else wants to make an announcement, they can let me know. And then we'll get underway with the exciting speakers for today. So this is a symposium that's being put on, by members of the International Compositae Alliance. And we also meet informally other weeks to have journal clubs and organizational meetings. If you'd like to get involved, feel free to reach out to me or to Carol by email, and we can put you on the list for that. We've had some exciting journal clubs recently, and there's some really nice papers coming out about Compositae that we'll be discussing soon. We are in the process of formalizing membership for TICA, and so you should receive an email soon with a link and a form that you can fill out to officially join TICA, and you can make a profile and connect with other researchers from around the world. For this symposium, we also have a link, which Carol will share, to sign up to give a talk. So there's still some spaces available, and we'd like to also make this as international a symposium as possible. So if you want to give a talk and you don't necessarily want to give your talk in English, what we can do is we can record the talk beforehand, and we can work together to add subtitles. And we are totally open to having symposiums that are in many different languages. It would be kind of fun and interesting to do. So if you'd like to sign up, please do so, and you can indicate there what language you'd like to give your talk in. Are there any other announcements that anybody would like to make? No? Okay, so then without further ado, let's get it underway. So first up, we're gonna have a nice talk by Erika. Erika Moore is a graduate student at the University of Memphis in Jennifer Mandel's lab, where she's studying systematics and phylogenomics of the genus Pachyra in the Sniciani. And today she's going to give a talk about a side project that she's working on. So let me make her a co-host. And she'll
I think I'm good.
Screen. You're already good?
Mm-hmm.
Okay. Then go ahead.
Okay. So hello, everybody. So today I'm going to be talking about a side project I've been working on with Carol Siniskalki and Jennifer Mandel, where I reevaluate the genetic diversity and structure of Helianthus verticilitis after the discovery of 2 new populations. And so to start off, Helianthus verticilitis, also known as the world sunflower, is a federally listed endangered sunflower species found in the southeastern United States. It is a diploid perennial that reproduces both sexually and asexually by rhizomes. In addition, it is self-incompatible and insect pollinated. It is morphologically distinct from other species of Helianthus by having rolled lower leaves, yellow disc florette corollas, and then a prominent midvein and short petiole within the leaf structure. It was originally discovered by Samuel Bain in 1892 in Tennessee, and it was not found in nature again for over 100 years until 1994, when a population was discovered in Georgia. A few years after that, in 1997, another population was discovered just 4 kilometers west of the Georgia population in Alabama. The following year, another population was discovered in Madison County, Tennessee, which was only 10 kilometers northwest of the original collection site. And then finally, in 2006, another population was discovered in McNairy County, Tennessee. Upon further inspection of these populations, researchers realized how small and distant these populations were. And so there was a call for population genetic work to aid in conservation efforts for this species. And so then this led to multiple studies performed by the now Dr. Jennifer Mandel that aimed to see how similar Helianthus verticilitis populations were, as well as how similar they were to closely related sister taxa. And so what she found out was that the Helianthus verticilitis populations do appear similar and they do cluster together. However, the Tennessee populations separate from the Alabama and Georgia populations. And so this makes sense just given how close Alabama and Georgia populations are, given they're only like 4 kilometers apart, compared to the Tennessee populations, which are much further. When she compared the Helianthus verticilitis populations to 2 closely related Helianthus species, Helianthus angustifolia and Helianthus verticilitis, she found that the species are distinct and they do appear to cluster separately. And so this was important to note since previous researchers had predicted that Helianthus verticilitis was a hybrid between Helianthus angustifolia and Helianthus verticilitis. And so with the help of those studies, and then some additional studies done somewhat around that time, we can conclude that Helianthus verticilitis is unlikely to be a hybrid, and that the Tennessee populations clustered separately from the Alabama and Georgia populations, indicating that there may be a little bit of gene flow. And so with the help of those studies, Helianthus verticilitis was then listed as federally endangered by the United States Fish and Wildlife Services in 2014, which gave legal protections to those 4 populations. Then a few years later, in 2017, another population was discovered in Marshall County, Mississippi. And so this population was collected by botanist Daryl Brandon. He actually spotted the collection while driving down the highway because this population's on a roadside. And he reported that it contained a clump of roughly 150 healthy stems. It was not flowering at the time of collection, but he was able to ID it, give it the whorled leaves, the prominent midvein, and then the short petiole in the leaf structure. He ended up collecting 13 samples from multiple clusters of plants in order to appear, and make sure they appear distinct in order to minimize collecting from clones. And after that discovery, I started doing some population genetic work to see how this Mississippi population compared to the other known populations. But then I had to start all over because in 2019, another new population was discovered in Franklin County, Virginia. And so this population was collected by plant ecologist Chris Ulrey. He found 2 distinct patches that were only about 30 meters apart. And given that it's a pretty close range for gene flow, we ended up treating it as one population. But Chris Ulrey ended up collecting 8 individuals, 4 from each patch, and then sent the tissue samples to the University of Memphis for me to research. And so the discovery of these 2 new populations, I wanted to ask, how similar are the Mississippi and Virginia populations when compared to the other known populations of Helianthus verticilitis? And then how do the levels of genetic variation compare? I also wanted to ask how much population genetic structure is there? And then are there any geographic barriers to gene flow? And then finally, what do these results actually mean for conservation? And so to address these questions, we wanted to genotype these newly discovered populations in Mississippi and Virginia, and then compare that data to the previously known and genotyped populations. But to do that comparison, we had to first standardize the genotypic data from Mississippi and Virginia to the older studies. And so to do this, we genotyped 3 individuals from the early 2006 collection, 2 individuals from Alabama and 1 from Georgia, as well as 3 individuals that are resulted from a cross between individuals from Madison County, Tennessee and Alabama. These individuals are cultivated in a garden at the University of Memphis. Using those 5 populations, we then ended up doing DNA extractions, ran PCRs using 13 nuclear microsatellite loci that are proven cross-transferable from Helianthus annuus, and then we genotyped those at the University of Tennessee Health Science Center here in Memphis. And so then after that, we then had both the Mississippi and Virginia populations standardized and ready for the next step. Given that Helianthus verticilitis is known to be clonal, we still wanted to assess clonality within the 2 populations, since having multiples of the same genotype could influence statistical analyses. And so by doing so, the sample size ended up decreasing in both populations from 13 to 10 individuals in Mississippi and 8 to 2 individuals in Virginia. But now that the data only contains unique genets and is standardized, the Mississippi and Virginia populations are now ready to be compared against the previously known and studied populations. And so to address the first question, we ended up doing it in 2 parts. So the first part is we wanted to look at how similar the Helianthus verticilitis populations were. And so we ran a principal coordinates analysis to see where individuals cluster together in order to assess their similarity. And so the first thing to mention is that there's a large cluster on the top left of the PCOA. And here we can see that there's 4 populations, Madison County, Tennessee, Georgia, Alabama, and Virginia, all clustered together. indicating that they are genetically similar. Next, we see that there appears to be 2 additional clusters of individuals from McNairy County, Tennessee, and then also Mississippi, meaning that both the Mississippi and McNairy County, Tennessee populations are less similar to the remaining 4 populations. Given that the Virginia population is a part of the first cluster, this indicates that the Virginia population is more similar to the previously known populations than the newly discovered Mississippi population, which forms its own cluster separately. One thing I do want to note is that previous studies had shown that the McNairy County population was the most genetically diverse population, like when tested previously. And so that's not necessarily new to this study, but what is new is that, or is where Mississippi and Virginia populations lie within this analysis. And so for the second part of question 1, we want to see how the levels of genetic variation compare between populations. And so to do this, we calculated measures of genetic diversity for all 6 populations. And so I can spend a lot of time discussing what this table is showing, but instead I just want to highlight 2 genetic diversity measures that really address the question at hand. The first being the number of private alleles. The Mississippi population appears to have the highest number of private alleles out of all populations tested. Therefore, there may be a lack of gene flow between the Mississippi population to the other known populations. Next is the inbreeding coefficient. Both the Mississippi and Virginia populations have negative inbreeding coefficients, meaning that there is a potential heterozygote excess and potentially little inbreeding within the populations. And this makes sense since some studies have shown that clonal populations tend to have a higher number of heterozygotes if heterozygosity is advantageous in that area. We do want to place caution though on the results of Virginia population's genetic diversity measures, since having such a small sample size could also influence statistical analyses, meaning the results may not fully represent the entire population. But even so, the results so far have shown that the Mississippi population appears to be the most diverse and distinct. And then Virginia is not quite as diverse, but potentially lacks gene flow with other populations. And so for the second question, this one's also going to be in 2 parts. And so we wanted to know how much population genetic structure is there. And so to do this, we end up doing a pairwise FST comparison. And so pairwise FST tells you how genetically different 2 populations are from each other, given a range from 0 to 1. Values closer to 0 show that they are more genetically similar or identical, while values closer to 1 indicate that the populations are not as similar or they're more distinct. And so I color coordinated the table to show values closer to 0 appearing more green, while values closer to 1 will appear more yellow. So the first notable difference is that the Mississippi and Virginia populations appear the most yellow and therefore have the highest highest pairwise FST values, indicating that they are the most different when compared to the previously known populations. But not only that, the Mississippi population has the highest pairwise FST value for all populations tested. And so this supports all our other findings so far about the Mississippi and Virginia populations being genetically diverse, especially the Mississippi population being the most diverse. And so for the second part of question 2, we want to see if there are any geographic barriers to gene flow. And so to address this question, we use the program barrier, which uses a geographic distance matrix and a genetic distance matrix to predict where potential geographic barriers are between populations. And so by running this analysis, 3 barriers were then shown to be significant. And so using those barriers, we then predicted what potential natural barriers could be separating these populations. And I just want to clarify that these are just speculations and that they have not actually been tested or confirmed. But the first barrier that showed up in this analysis was the barrier between the Mississippi population to the remaining populations. And just given where it is, we predicted that maybe the Hatchie River in Tennessee and Mississippi. The second barrier that shown up was between the Virginia population to the remaining populations. And so we predicted that maybe the Appalachian Mountains. And then finally, the third barrier was between the eastern populations and the western populations. And we believe that natural barrier may be the Tennessee River. And so even though these are just speculations, it is still interesting to see what natural barriers may be limiting gene flow between populations. And so finally, we want to ask, what do these results mean for conservation? And so first, our data shows that the Mississippi and Virginia populations appear to be highly clonal, and they also harbor novel alleles that are genetically distinct from the previously known populations. The Mississippi population would be a prime candidate from a conservation standpoint, just given how genetically diverse it is. Unfortunately, the Mississippi population is unprotected and is continuously mowed or sprayed annually. And since its discovery, the plant has not been seen flowering, which is a concern for the health of the population. In the Virginia population, only 2 unique genets were identified out of the 8 originally collected individuals, showing that the Virginia population is mostly formed by rhizomes, and therefore is potentially under the most concern for conservation. And then finally, these populations could be useful for sourcing individuals for both seed bank conservation or for planting in restoration or management areas. So an investigation of the potential for local adaptation of these newly discovered populations is possible. Any transplant studies would benefit from monitoring fitness of individuals for several years. But even so, we hope that this work will aid in conservation efforts to protect these newly discovered populations to ensure that Helianthus verticilitis remains genetically diverse. And so I just want to end the talk with a brief mentioning of a somewhat recent adventure that we went on, where we did a land survey around the Mississippi population to see if there are any other Helianthus verticilitis populations near the Mississippi site. And we went and did this just because of how genetically diverse the Mississippi population appeared. We kind of speculated there may be additional populations that it is crossing with. And so we ended up actually finding 2 additional populations that are potentially Helianthus verticilitis. I say potentially just because these populations were morphologically highly variable, both in leaf venation and petiole length. And so we want to do some genetic work to see where it fits with other verticilitis populations. And then also we want to test the other Helianthus species around it. But if you are interested in this land survey, I've attached a QR code in the top right to a blog post I made on my website where I go into greater details of the trip. But with the discovery of new populations, it's exciting and suggests other yet undiscovered populations may be found. So we recommend further searches for populations, focusing around the areas with suitable habitat and then also near those newly discovered populations. And so then with that, I want to thank my co-authors, Jennifer Mandel and Carol Senskalski. I also want to thank Jorge Gomez for his help with lab work, and then Daryl Brandon and Chris Ulrey for discovering the populations for me to actually have something to work on. And then also Tom Cunningham at University of Tennessee Health Science Center for genotyping the data. And then with that, I'll take any questions.
So Erika, we have a question in the chat from Dr. Ed Schilling, who asks, did you try to estimate the time since divergence among the 3 clusters?
We did not. I actually wouldn't, I'm not a population geneticist necessarily, so this is kind of new for me, so I'd have to look into different ways to do that. But as far as I know, we did not. Any other questions? Oh, okay. Okay. And so
if you want to read it out loud.
Okay. Yeah, and so Mauricio has asked, have you tried searching for more populations? And we have not yet. It's kind of limited because once we found these new populations, it was in 2019, and so it was already post-flowering. And then in 2020, we had the world happen. And so we couldn't go out to many places, but I think we're still planning on looking for more populations when we can. We could ask Chris Ulrey back in Virginia, but it's a little bit farther from where I am, so we'd need help on that one. And then the other question, to what degree has the species been taken into cultivation? So this was just a cross that was made between Jennifer Mandel. So she ended up, or she was growing the plants, I believe, for a hybrid experiment, trying to make sure they weren't actually, like, couldn't produce viable seed. But then with other species, I should mention. But then with these populations, then she was able to actually cross the populations to then produce the seed for the botanic garden. But there's, as far as I know, it's only just been distributed to local gardens in Memphis.
There is an effort, though, across the state, which is who we were working with when we went on that collection trip, to source seeds and find potential locations in West Tennessee where they can do sort of like experimental sort of plots of putting verticilitis out into nature. So there's an effort with the Tennessee Rare Plant Conservation Alliance that is sort of trying to gather everybody up. So like the Botanic Garden here in Memphis, as well as some other nature centers and Strawberry Plains in Mississippi. So there's a lot of interest in this species. Everybody loves the sunflower.
Thanks, Erika. If there's some more questions, and maybe if you'd like, you could write out answers to those in the chat while we continue to go. And then also, if we have some more time at the end, we can come back and ask more questions.
Okay, so, awesome. Thank you, guys.
Thank you. So that was a great talk. And next we have another another shorter talk from Gabriel Johnson, who is a laboratory technician at the Smithsonian Institution associated with the National Museum of Natural History. And he is an expert in all the different ways of obtaining DNA from plant extracts in order to get great data, and is going to help us understand some of the common misconceptions about extracting DNA from plants. So with that, take it away, Gabe.
Hello. Let me get my slideshow up here. Yes, so common misconceptions about tissue preservation for DNA. Yes, extracting DNA is always fraught with a lot of confusion because just like plants are so diverse, their extracts are equally diverse with all kinds of interesting things that co-extract with the DNA. And just depending on where the DNA was collected, how it was collected, or how old the plant was when it was collected, you can get a range of different results in your DNA extractions. And so there's lots of anecdotal evidence around different ways of extracting DNA that is best. And there's not that much hard empirical evidence because usually the method section of a lot of these papers are citing the method section of another paper that cited another method section of another paper that cited another method section that goes down very far and usually doesn't have a lot of details because most of the details of the studies are saved for the actual results and discussion rather than the nitty-gritty of how they collected the DNA. But nonetheless, over the years, there has been efforts around the world to get some more firm empirical data on just exactly what's happening in that tube when you extract DNA. And yeah, I've sort of compiled that here, and I'm going to go over some of the more notable misconceptions that or eyebrow-raising things that you may discover from the DNA extraction literature for plants. Yeah, so I guess my first thing I'd like to say is this famous Barbasol shaving cream bottle with dinosaur embryos in it is not real. I think you all know that. But also, unfortunately, the cryopreservation technology to fit an entire negative 190 preserved environment in the size of a can for 7 days straight is still actually science fiction. If you see this and it costs $341 US.47, it is not real in spite of that ridiculously high price. So because we can't really go out and preserve tissue in liquid nitrogen as much or as easily as we would wish, we have to make do with the other technologies that are available, which in many ways are just as good and sometimes better than if you were able to theoretically collect all of your plant tissue in liquid nitrogen in the field. So we're going to talk about herbarium specimens and what happens inside those leaves when they're collected and pressed and how that affects the DNA. And then talk about the gold standard in our community, the silica gel desiccation. And then I'm also going to contrast that with a seldom used but perfectly legitimate desiccation method in ethanol, and then go on to explain how really we can use both. Yeah, so when a leaf is removed from the plant, it immediately begins senescence. And if you took plant physiology, you know there is a whole pantheon of hormones that are released, from ethylene to jasmonic acid to abscisic acid, that begins a program of genetic processes to take that leaf, break down all the usable stuff, put it in the phloem, and send it to the roots so that the plant can use it later. It is not random death. When a plant senesces, it is a highly organized, meticulous process. And that involves the autolysis of DNA. The plant will immediately begin to break down the DNA at those little nucleosomes where each DNA loops around the histone. And breaking down the DNA into its nucleic acids makes it easier to accumulate those nucleic acids in the phloem to send elsewhere. Also, the plant does not want this dying leaf to become a gateway for parasitic infections, bacteria, and fungi to take over and kill the plant. So they want to make this dead leaf a firewall for anything else to come in. So those peroxisomes you learned about in TEM class long ago, they start to make a lot of reactive oxygen species. And there's phenyl oxidase reactions that occur to create these dark pigment substances. And a lot of them have antifungal properties. Unfortunately, a lot of them also have anti-DNA properties. And a lot of these reactions will also continue to break down nucleic acids and things in the cell. So that is why it is best, or you probably have found, it's easiest to get DNA out of a leaf once it has been efficiently dried. There is a significant difference between letting a leaf just wilt and senesce on its own versus using an oven to hot air dry it. Drying tissue faster is a race against time to preserve the leaf in a dry state before these senescence, this apoptosis, this programmed cell death can occur. Cell death and these responses to wounding occur at a certain rate, and we can dry the plant faster than these enzyme reactions can occur and prevent the DNA from being degraded enzymatically. And that is why historically you can get fairly good DNA from a herbarium specimen rather than if you collected the leaf and put it in your pocket and let it just get all wilted and dry by itself. That increased speed of drying is a good thing. So yeah, faster drying means better DNA. And here we see the Global Genome Initiative at work, and Vicky Funk making these pressed specimens in a very organized manner of making a pressed herbarium voucher. And the woman you see on the right-hand side with that bag of silica gel is taking silica specimens. And you also see a woman in the middle with cryo tubes taking fresh specimens and dropping them directly into a tank that is cooled with liquid nitrogen to -180 or -190 Celsius. And this is ideally how we would collect everything. But unfortunately, not everything grows in a greenhouse where it's so convenient for pressed specimens to be made and cryogenic and silica samples to be preserved. And that's why we have to get creative. Now, silica gel has always been our go-to standard since the 90s for drying out leaves. But what is often not totally understood by the entire scientific community is that just as easily as the silica gel can dehydrate the leaf nice and quickly to prevent DNA damage, that silica gel can also become saturated with moisture, and eventually it will stop doing its job. It only works when it hasn't changed color. When it changes color, it is fully saturated. And then humidity from the air will begin to pass through that polyethylene bag into the silica gel and into the leaves. And then the leaves will be not as dry, not as wet as a fresh leaf, but it will be significantly higher in humidity than it was when the silica gel was its original yellow or blue color. And a leaf that is at the ambient humidity of a room is actually significantly harder to obtain DNA from than a leaf that is completely desiccated to the humidity of fresh silica. When silica is still fresh in its original color, the humidity inside of that baggie is as low as 10 or 20% relative humidity. But once it has changed color and become fully saturated, the ambient humidity in the room, which is 30 or 40% relative humidity, that is enough to make the polysaccharides in the cell walls a little more flexible, and they won't pulverize as easily into a fine powder in bead beaters when you try to extract the DNA. And as with most things in life, the beginning of a process is a foundation that really needs to be as best as possible. And it doesn't matter how good your downstream DNA extraction methods are, if you can't get a good pulverization, a good tissue powder to begin with, nothing else will purify the DNA. You can't get DNA from a powder that is fragmented and not fully fully and efficiently homogenized. So yeah, making sure that your silica gel is always completely fresh and is not fully rehydrated is a key element. You often need to change the silica gel after making collections. And that's why a lot of researchers are getting away from using these polythene bags with tissue on silica contact, especially for Asteraceae, due to the thin nature of many of these leaves. It's perfectly fine to put the tissue into a coin envelope and put those coin envelopes into a container of silica gel. And that makes it easier to change the silica gel with fresher, dry silica gel as needed. Yes. And just as a tangent, I'd also like to say that those bags and coin envelopes of tissue, if you sit them on your office desk, even if it's in a very institutional building with very few windows, even the light from your fluorescent tube lighting above, that light can really harm the DNA inside of those leaves. And there is significant empirical data to suggest this, that the DNA that you extract from your silica dried leaf specimens, if you leave those sitting under your office lighting for a month, you will have fairly significantly and demonstrably increased the amount of fragmentation of that DNA. So it always is important to make sure once you've collected your specimens and you're done working with them for the day, you put them in a dark closet or drawer in the office or laboratory. Yeah, and so I like now to talk about the idea that you can use ethanol and other alcohols just as effectively as silica gel. And just to let you know, you can get all kinds of plants in alcohol on the internet for decorative purposes. But no. Here, this desiccation in alcohol is not to preserve morphology as we do in our wet collections. No, rather it is to preserve the DNA inside. Now, depending on the thickness and the amount of lipid in the cuticle, certain leaves will desiccate faster in ethanol than others. And for those that do desiccate in ethanol, sometimes it's better to use the ethanol than the silica gel. Just to show you how it works, here is a little experiment I did once with a collard leaf. I sort of removed all of the big, thick stem, big, thick vascular bundles, which don't really have a lot of DNA per unit area, and then ripped it up into about 1 by 1 centimeter pieces that are easy enough to put into a tube of 95% ethanol. Now, mind you, that needs to be 95% and above. Basically, any kind of laboratory grade ethanol. If you dilute it down to 70%, as is often done for pickle collections, that does not preserve the DNA as well. The key is to stop any of the internal enzyme processes of wound response and apoptosis, senescence. To stop that abruptly and to remove water away from the cell wall and the DNA, as the hydrogen bonding of water, that Brownian motion of molecules and the bonding and breaking and bonding and breaking of hydrogen bonds from water, that is what is so destructive many times for the DNA. And if it's above 95% ethanol, that is the most effective concentration. Now, if you ever are going to go this route, you need to remember that ethanol is a solvent for ink as well, and that writing the name of the organism and your collection information with pencil on paper to insert into the ethanol tube is the best way to go, since the pencil lead won't dissolve away in the ethanol solvent. But also label again with ink, but make sure it's one of those fancy Sharpie markers that is quote unquote solvent proof. You know, it really isn't. It still kind of runs when ethanol touches it, but it doesn't run off completely. So get one of those solvent proof Sharpies and also label the outside of the container. It's true no matter what stage you are in your scientific career, you can never label too much. More labeling, the better. Yeah, so ethanol, like silica gel, when the silica gel is fresh and not spent, will not only preserve the DNA, but it also makes the cell walls brittle. And that brittleness increases its efficiency in grinding and creating a better ground, more finely powderized tissue is key to getting a larger DNA yield. And when it's really hard to collect these plants in the wild and you don't have much leaf tissue to begin with, every extra 0.00 nanograms of DNA is essential. So ethanol dehydrates cells and preserves the DNA just as well as silica gel, but it has an extra benefit in that the brittleness that it renders to the cell wall is not as reversible. As I said, when your silica gel changes color and is spent and hydrated, the humidity of the silica gel becomes the same as the room. And room ambient humidity makes leaves a little more flexible enough to resist grinding completely. While ethanol must remove some kind of substance from the cell walls, which sort of renders them permanently brittle, that even after the ethanol has evaporated and went away, those cell walls will always be a lot more crunchy and brittle and will always make a finer powder. Not only do they have make irreversible changes to the cell wall substances, ethanol also irreversibly destroys a lot of nucleases. One misconception about DNA extraction is that you don't have to worry about any of the DNases that are, your body is just covered with. Your body is always trying to protect itself from viral infections by sloughing off all these skin cells and sweating out all this sweat that contains lysozymes but also nucleases that destroy viruses on contact. And they can also sort of, your skin cells can slough off into your samples. And these nucleases, by and large, need magnesium and divalent cations to be effective. However, there are suites of families of nucleases that are completely unaffected, independent of magnesium. And so we put EDTA into our lysis buffers, believing that it's going to chelate all the magnesium and prevent any DNA's enzymes present from harming the DNA. And that is true, but depending on the species of plant, they may have their own enzymes that are perfectly preserved in silica gel that once you rehydrate it in the lysis buffer will become active again and will start to digest your DNA within minutes. And that sounds like a horrible boogeyman that I just described, but it's true for certain groups. Unfortunately, plants, like all living things, are diverse. And the amount of these magnesium-independent nucleases, they're in different concentrations in different plants at different organs and in different stages of their development. And for your particular group that you study, this may not be a big deal. But for some plants, this is a big deal, where those nucleases need to be inactivated with something like ethanol, because the EDTA just won't cut it because they're magnesium-independent nucleases. Here's a study that was done comparing silica dried versus ethanol treated. The top two images, A and B, you see, is a toluidine blue stained thin section, well, thick section, and a TEM thin section of something that was treated, that was dehydrated just with silica gel. And then below, it's where you've dried it out with ethanol first. And you can see the cytoplasm is really grainy and crazy with the ethanol treatment. Like, that is one ugly TEM image. And that's because all the proteins got denatured by the ethanol dehydration, while with the silica, yeah, it dried out, but upon rehydration in the fixation buffer and such, it's they're not as, it's a more elastic, it's a more it's a change that is not permanent. So now that I've introduced you to this idea of preserving with ethanol, I'd like to say that ethanol is not like a replacement for silica gel. It's just one extra preservative substance that you should have in your toolkit that you can use to preserve tissue for DNA extraction if it is needed. And surely, and many papers have described, you can just dry things in silica gel. It's just, if you need to, then also do a secondary dehydration in ethanol and let the ethanol evaporate, and then pulverize the tissue for DNA extraction. That you can use both silica gel and ethanol. Silica gel in the field and ethanol in the lab. But regardless of how much ethanol treatment is used on your tissue, bear in mind that in many cases, the ethanol makes the proteins coagulate. You see here how when the cytoplasm, the TE image in figure D, how all those proteins sort of turned into little tiny granule things. That granular protein sort of mummifies the DNA. It sort of coats the DNA because the nucleus, we think of that as, you know, where all the DNA is in the cell. That's true, but there's equal parts protein to nucleic acid in the nucleus. There is a lot of protein and transcription regulating things and enzymes in the nucleus that will coagulate around your DNA. And so after treating your tissue with ethanol, oftentimes that protein needs to be enzymatically removed with proteinase K. Not very much. I mean, we're not digesting through a whole rat's muscle here. It's just the protein that's in a plant. So it's a smaller concentration of proteinase K. And if you want to save money, there's this stuff called pronase E, which is a cheaper, less effective, but for plants, perfectly effective enough protein digesting enzyme that you need to add to your lysis buffer. Yeah. Yeah, so I'd like to thank you for your attention, and yeah, let me know if you have any questions.
Thanks, Gabe. I think we maybe just have time for one question, if that's okay. And then if there's more questions, you can put them in the chat and Gabe can respond there and include everybody since we're all interested in the answers. So maybe Morgan has a question, which is, he says, great to see the ethanol tricks demonstrated, Gabe. Do you have any recommendations for traveling with or transporting 95% ethanol?
Yeah, I work in the lab. I don't have to deal with all that horrible stuff. But yes, transporting stuff in ethanol is a problem because it's a volatile, explosive liquid. Fortunately, you don't have to bring it with you in your suitcase. You can just sort of buy the ethanol there in-house, which at least so half of the transportation means you don't have to have it. You can come to the country without the ethanol, buy it there and use it. Unlike some people who have to bring suitcases full of nothing but silica gel because they can't get it in the country. But no, you can preserve stuff in silica gel. You don't have to transport things in ethanol. To get the value of the ethanol, to make the cell walls more brittle and to protect the DNA from nucleases that are magnesium independent, that can happen in the lab. So preserve it in silica gel. And then once you bring it to the lab, then you can use the ethanol. So it doesn't really have to be pickled in ethanol in the field.
Great, thanks, Gabe. So for our last talk of the day today, we are excited to have Robert Edwards, Dr. Robert Edwards. We all know him as Bort, and he is a postdoctoral research associate at Yale University in Walter Yett's lab. And he's an expert in using big data to study daisies on a global or continental scale, and going to give a very interesting talk. So with that, take it away, Bort.
You're muted, Bort.
Thank you. All right, so I was just saying thank you to everybody for turning up. And this is going to be a fairly quick sort of mushing together of two concepts that could sort of take a whole day of working with in their own right. One of which is GBIF data and sort of how do you deal with that? And then if you have managed to deal with your data, what can you do with it in terms of sort of some gross patterns of trying to establish where sampling efforts might be lacking, where we might be concentrating our work in going forward to look at plant diversity and compositae diversity. So,
hey, Bort.
Yep.
We're seeing your presentation view, just so you know.
Okay.
So maybe you might need to change the view to use slideshow at the top, or if you don't, I mean, you can also try that.
Well, that's not working well.
Or swap displays or mess around with that. You might need to exit and try it.
Okay. Now both my screens have gone dead. This is good.
Yeah, that's perfect.
Okay.
Oh,
Is that better?
No, whatever you did a second ago, if you go down maybe and hit the presentation on the bottom right, try that.
Yep.
Looking for a second there. Yeah, try that.
Yeah.
That looks perfect.
Is that better?
Yeah, great.
Okay, I think I might have had dual screen issues there. You're probably seeing the wrong screen. Okay, so, all right. So understanding diversity is very important. It's tied in with the fitness and the survival of the human race. We depend upon diversity of plants and animals for survival, for the planet's survival, and as well just for sort of our jobs as biodiversity people and scientists. But getting a grasp and a handle on quite how diversity is made up is quite difficult. This is because it varies considerably, not only through time, so through epochs, geological epochs, as the continents move, as the climate changes. It varies through space, as you drive across a continent, as you move between continents, as you go up and down a mountainside, it'll change. And it also varies at scale. So the number and diversity and density of organisms, plants that you find in your backyard is different if you were to draw a box around your city, draw a box around your state, or look at a global scale. So trying to get a handle on how diversity varies is quite difficult. And it's only really recently with these sort of very large collaborative data sets, diversity by diversity data sets becoming available, that we're able to start to sort of transfer between these different scales more easily and use data sort of at different slices. But this can cause some problems. So how do we get this data? This data exists, as we all know, in herbaria, in museums. And one of the ways to do this is to aggregate it all in one place and then serve it out to the people who need it. And I think we're probably all familiar with GBIF, which is the facility for collecting and serving spatial data. So this is aggregating all the collection records for plants and animals, but we're interested in plants, from institutions around the world. What might not always be quite so apparent, though, is that GBIF itself doesn't actually doesn't own the data. It doesn't even take on the data. What it does is it serves as a sort of a central node. When you query a species name, it will send that query out to the nodes under it, the museums, the local museum consortia, or the other repositories of the data, pull the data in, and then serve that to you as a packet. So this means it's effectively coordinating your search. If you ask for one species, it's asking all these different nodes, do you have this species? What collections do you have? And what are the records? And this is great. It works relatively well. You can query a global network of institutions and ask, do you have this species? But the problem is, when you're trying to pull in a whole lot of disparate data sources, you've got to try and sort of agglomerate them in a way that's useful to the user, bearing in mind that you're taking this data from a whole bunch of different sources. And these different sources might have different names for the same specimen, different names for the same species, depending on which taxonomy they follow. They might have a slightly different standard for the way they record or store their data. And so what GBIF is really doing is doing a lot of magic in the middle there. It's translating what's coming in from its outlying sources, compiling it, conglomerating it, and then serving it to you as a user. And without really knowing or with knowing what goes on under the hood, the first really interesting big question you have to ask is, do you want to use GBIF as a data server where you're effectively just saying, hey, go get this raw data for me, bring it back, and I'll look after it myself? Or do you want to use some of the value adding that GBIF does in that process to save yourself some time and effort? Because as GBIF is taking these records from around the world, it's doing things like running a taxonomic harmonization. It's saying, okay, so I have 3 names here that I think apply to one species, and it combines them together, and it will then serve those to you under what it considers to be a standard name. However, you as a researcher might not necessarily agree with that taxonomy, or might not want to use GBIF's idea of a taxonomy, in which case you don't want to use GBIF as a data curator. You want to use GBIF solely as a data server. And I think that is something that's very easy to overlook. And in my opinion, GBIF is its most powerful as a data server, and you start to run into more problems if you use it as a data curator. And partly this is because of its harmonization of taxonomy. That's where it does its main heavy lifting. And this is where it is pulling in different names from different sources and trying to reconcile those against its standard taxonomy. The problem with this is that it's a relatively opaque system. They're taking a whole lot of different plant lists, plant taxonomies. They arrange them in a hierarchy, and they slot the names into them, and they run it down through these lists to try and work out what they consider to be an accepted name. However, these algorithms are largely automated. And so you lack a lot of the fine scale taxonomic expertise that you might be wanting to use if you are a taxonomist, or in fact, using a name at the other end. So I would say that if you want to use GBIF data, you want to use the raw names that GBIF is pulling in from its servers, not the taxonomically aggregated names that it produces at the other end. The other thing that GBIF does, it does flag spatial issues in the data. So there's sort of 2 tranches of what GBIF does with the data. It tries to taxonomically standardize it, and then it flags anything that might have a problem spatially. The good thing about this is that GBIF doesn't really try and force too much of this down your neck. It will flag and say, this record has an issue. It looks like it's in the wrong country compared to where the collection record says it's from. It's falling in the ocean when it's probably not an ocean growing plant. These lat longs look suspiciously rounded, things like that. You can ask GBIF to automatically screen all those records out, at which point you are asking it to curate the data for you, or you can take that data wholesale. But relatively speaking, at least what GBIF does is provide you that spatial data relatively raw, and it gives you the ability to clean it and use it as you see fit. So what we have been doing to try and get an idea of the diversity of vascular plants worldwide, and I will get to why this is complicated soon, is that we've created our own master taxonomy. So we started out with the Leiden Catalogue of Vascular Plants, which was published late last year, early this year. And then we augmented that by running it against the World Flora Online and the Plants of the World Online, identifying names that didn't match between the two, and then trying to reconcile those in the World Flora and the Plants Online that were not matching to the Leiden Catalogue. And that allowed us to add in a whole lot of synonyms and orthographic variants and typological errors, and blow out the list of names to about 1.3 million names. So we then took our GBIF, the GBIF data for all the vascular plants, and compared the number of names that GBIF provides, which is 890,000 names, and ran that against our master taxonomy. And as you can see, we ended up pulling out about almost 600,000 names that have records in GBIF that our master taxonomy agree are either valid names or recognized synonyms. But that still left us with almost 300,000 names, almost 1/3 of the names didn't even match. So despite having pulled together and mushed together the two largest online databases of names, 1/3 of the names that GBIF recognizes in its database don't have a match in our taxonomy. If we sort of drill down into what might be going on here, or at least what the sort of this mucky, unmatched end of the name spectrum is, we can see that if we look at the number of records per name, so these are species, allegedly species, that GBIF only has pulled out a single record for, we have about 67,000 names. So there's an awful lot of singletons floating around out there. And we know from previous that these are quite possibly false species. They don't exist, they never existed. They might be typographical errors that are just too hard to resolve through a simple fuzzy match, or things that were discovered once and never seen again. And so the question then begins sort of, how far down this rabbit hole of cleaning and trying to reconcile these names do you want to go? As you can see, this is a very long tail. There are still 6,000 names often that don't match between GBIF and our master taxonomy that have upward of 1,000, up to almost 700,000 records associated with them. So some of these names, a lot of these names that we're not matching are obviously very widely used, and presumably many of them are valid. So somewhere in there, there's a sort of a tipping point at which you have to either acknowledge that you're going to ignore these names and they're just not going to be pulled out of GBIF, or you have to go through and reconcile them. So to some extent, we can do that. A lot of these mismatches, it turns out, are things like orthographic variants. So that's where you have a slight misspelling or difference in spelling. A lot of this happens when you try and change between Latinized versions of names and English versions of names, and there are different ways of resolving that. There's also changes between the gender of a name that often ends up with slightly different spellings. For example, here it's just a transcriptional error at some point, literalis spelt with 1 T or 2 Ts. And so you can run through an algorithm and ask it to fuzzy match. So your names that you submit from GBIF, you run a fuzzy match against your master taxonomy, and it pulls out the best match that it can. The problem with that is that it'll pull out the best match, which doesn't necessarily mean it is the match. You can find it pulls out something that looks kind of similar, but is very, very different. So you still have to go through and verify these all by eye, which takes an awful lot of person hours. The other thing that we found a lot of are autonyms. This is where a subspecies, usually where a subspecies has been erected, but doesn't include the type of the species. So a sibling subspecies automatically erected sort of by exclusion, I guess. It hasn't been officially described. There's no official author. The author is assumed to be the author of the original taxon. But these names sort of exist sort of by presence of something else. And so a lot of these names have never actually been published or described, and so they're not going to be picked up by checklists or names of plants that are actually looking at entities that have been used in the literature. So a lot of these names are floating around. And then we have all sorts of issues with author abbreviations where people have entered the data in different ways in different databases. And that is a whole other headache unto itself. So some ways around this is to use fuzzy matches and then to human screen these to try and match up names with their actual, their matching name in the master taxonomy. Okay, just to say, as I said, we don't really want to worry too much about spatial issues for the level of data that we're going to be talking about a little later. Just to say that there are definitely spatial issues. You get plants, as you can see, if you map terrestrial plants, you get plants that are growing in the ocean where they shouldn't be. You get often mirror images of the continent if you're looking at a particular continent where the lat longs are inversed. So these are other things that you have to clean as well. There's a whole suite of tools. But these relatively, you can run them automatically. They, as long as you're sort of willing to throw some babies out with some bathwaters, this is something that can be done on an automated scale relatively efficiently. It takes less human supervision than going through lists of names. Okay, so we have our data now. We've cleaned it for name issues, for taxonomy. We've consolidated under our master taxonomy. We've cleaned up our spatial issues. Perfect. We've got great data, right? So we have solid data for all of Asteraceae across the world. And we sort of look at the map. So this is just mapping the number of species by country globally. And as you can see, we've got variation, which is great. We don't want to have exactly the same number of species everywhere. That would be boring. We've got sort of high levels in Asia. Australia is well represented. South America is pretty good. North America. Africa is a little bit poor. And so is the Middle East. But maybe this is just because we get fewer daisies there. I mean, that doesn't sound really particularly intuitive for Africa, but maybe that is true. And definitely you could go ahead and sort of publish a map like this and say, hey, look, we just don't find as many daisies in Africa. They're not so great there for whatever reason. But if you start to pull down other ways of slicing or dicing this data, you start to see that maybe this isn't actually the case. So if you look at the number of records per country, so we're now getting an idea of the sampling intensity. We see that, again, the same countries that were coming up as having a large number of species are coming up as having a high number of records. And we see a similar pattern that Africa and the Middle East is underrepresented in terms of records. So this starts to sort of make us think that maybe it's that we just don't have enough data or the right data coming out of some of these countries that are poorly represented. If we look at records per species, so this is digging even a little bit further down. So, okay, let's assume that we haven't discovered all the species in some of these countries. We haven't gone out there a lot. Are we at least sort of collecting a fair number of records for those species that have been found? And again, we find a similar pattern that our sort of our first world countries, countries with the resources to go out and hunt locally, like Australia and some of the Asian countries, North America, Europe comes up very strongly, is well represented. People are going out and resampling species that have been already found, but Africa and the Middle East is falling very short. So it looks as though our sampling intensity in these areas is just really not what it should be. Interestingly as well, that we see sort of on the western edge of South America and a little in the middle, we also start to see that maybe we could be doing a little more in terms of records per species. That, or we just have a lot of rare species as well. So that's something that you would have to try and tease apart there. And then if you really want to try and sort of factor in, so maybe it's more a function of can you just not get into these countries because they're so large? There is certainly some of that as an effect. The larger the country, you sort of can dilute your sampling effort. And all countries seem to sort of suffer from some degree outside of Europe for having relatively few records, relatively for area. But again, we see that these places like Africa, even countries that are modest in size, are not very well represented at all in terms of records. And so it looks as though we really do have this, it's a Wallacean shortfall. This is not a novel observation, but it's something that we're trying to quantify and try and work out where are the areas that we maybe want to start to sort of focus on to better understand the flora at a global scale. To do this at a continental scale, I'll just throw this up quickly. You can also do this, sort of translate it to a continental scale. You can aggregate the diversity of this case. This is still Compositae in North America down to grid cells. And then you can do a rarefaction curve, which is a little bit like a saturation curve. You sample from each grid cell, see whether it's a species that hadn't been sampled out of that grid cell before, return it, repeat, and see at what point you start to keep recovering the same species over and over again. That's where those curves flatten out. And as we can see here, the flat curves on the left-hand side, the curves start to flatten out sort of from those mid-latitudes if you color the graph by latitude. Those mid-latitudes seem to be relatively well sampled. As you move southwards into Mexico, we see those lines are not flattening out. It looks like we don't have saturation. We have species numbers there that probably aren't reflective of the actual diversity. And the same going north into northern Canada. And if we do the same sort of an analysis looking at longitude on the right-hand side there, we see that the West Coast is relatively well sampled, well saturated. But then we start to get into the mid-latitudes, and maybe this is because we have mid-latitudes in Mexico, which is skewing these results again as well. But that sort of middle part, those middle latitudes also seem to be underrepresented. We haven't maybe collected all the species or collected as well as we could in those middle latitudes of the continent. So sort of using these sort of big gross patterns to try and work out maybe where we could be concentrating some effort in terms of looking for composite diversity globally and at a continental scale. So part of the work that I'm doing at the Yates Lab is to try and sort of get a handle on this. As I said, we're sort of at the moment trying to do this sort of for the global vascular plant diversity, get an understanding of diversity and where it is and how it's distributed across the landscape. And then we want to look at, I want to continue going with some of what I've been doing the last few years, as how extreme environments might be helping drive this diversity. But one of the remits as part of this work is to look specifically at the Compositae and use it as a test case to produce these diversity statistics and diversity maps. So I'm looking forward to sort of talking with a lot of you in the future going forwards who might be taxon experts particularly to do some bullshit testing of the maps as we start to produce them and see whether the patterns we're finding make some sort of intuitive sense. But generally the overall goal is to try and leverage this understanding of diversity. I think it's a grand goal. I can see many problems, but there is a large political component to this of trying to encourage countries to take better stewardship of their resources, to provide some sort of metrics to point to and say there are species here that we don't even know exist. There are species here that are unique to your country and that we need to start making efforts to find, protect, and look after them. So that's the work going forwards. Hopefully I'll have some more solid results coming along and be able to give another talk to you all in the future. Thank you very much. If you want pretty pictures that aren't comp related, I have Instagram and Twitter occasionally, and please reach out on email if you'd like to know anything about banging your head against large data sets and trying to clean them. Thank you very much.
Great. Bort is giving our last talk of the day. So now we have some time available for anybody to ask a question of any of the three speakers, or also if you want to make a comment, or if you have an opinion, or input from your experience researching Compositae, feel free to do so. And then you can also write in the chat if you'd like to. There's not any immediate questions. I'll just remind everyone that there is going to be a form sent out to formally join TICA, and we're getting that together now, and that the form is also available in this chat to sign up to give a talk. So if you have any questions about that, you can also email the TICAtalks email address that sends out the announcement for these talks and ask any questions as well. Or you have one question in the chat.
Yeah, that's a good observation from Alexander. So it's something that's almost unique to plants working with animal people now. It doesn't occur to them that you might have duplicates of collections. And so they often don't think to try and screen for duplicates. and from my previous North American Compositae work, duplicates are not even duplicates sometimes. So if they're sent to different herbaria, they can be entered slightly differently. You have different rounding of lat longs. So you can't just go through and screen for identical duplicates if you want to do a good job of it. You have to screen by collector and date, and then maybe a lat long to within a certain number of degrees, or round everything up. And that can be a real problem, as Alexander points out, if you're trying to do analyses that rely on density or number of records per unit area, then cleaning your data that way becomes very important. That is a huge issue. It's something, as I said, maybe because it's confined to plants, it's something that isn't well talked about or treated. People just screen for duplicates in a sort of pretty gross way. So yeah, it's a long and painstaking process to do it properly, and there's a lot of shortcuts out there that sort of try and avoid if possible.
All right. Well, I'll stop the recording and say thank you for everyone who gave a talk today and everyone who came. Thank you, and we'll see you in future TICA talks.